Hi everyone, myself Mrs. S. Indhumathi, Assistant Professor, Department of Microbiology, KSR College of Arts and Science Pavaman, Tiruchengood. Now I am going to discuss the topic microscopy. Microscopy is nothing but the science of investigating the tiny object which we cannot see through our naked eye, but we can see that object via microscope. In Greek, micron means small and scopios means to look. Microscope is an instrument for viewing objects that are too small to be seen by the naked eye. So, microscopy is the science of investigating that small objects by an instrument called microscope. Then, terms and definitions. First of all, we should know what is the principle of microscope. Microscopy is to get a magnified image, that is enlarged image in which structures may be resolved which could not be resolved with the help of our naked eye. Next, magnification. Magnification is nothing but the ratio of size of an object seen under microscope to the actual size observed with our naked eye. The total magnification of a microscope can be calculated by multiplying the magnifying power of objective lens with eyepiece. Resolving power. It is the ability to differentiate two close points as separate ones. The resolving power of human eye is 0.25 mm. It means that a human eye can be able to differentiate two different objects that are placed together or close with the distance of 0.25 mm. The light microscope can be separate dots that are 0.25 micrometer. In case of electron microscope, 0.5 nanometer apart can be separated as different dots. Then next one is limit of resolution. It is the minimum distance between 2 points to identify them as separate. It is calculated by AB equation. Resolving power is equal to wavelength of light in nanometer by 2 into numerical aperture of the objective lens. So, limit of resolution is inversely proportional to the power or resolution. If the wavelength is shorter, then the resolution will be greater. Working distance. It is the distance between the objective lens and the stage or specimen slide. The working distance decreases with increasing magnification. When you go for higher magnification, the working distance will be low. The next one is numerical aperture. Numerical aperture of a lens is the ratio of the diameter of the lens to its focal length. Numerical aperture of a lens is an index of the resolving power. Numerical aperture can be decreased by decreasing the amount of light that passes through a lens. So, first of all, light microscope. Light microscope was first constructed by Johnson & Johnson in the year 1590. In 1665, Robert Hooke developed the first laboratory compound microscope. Later, Kepler and Galileo developed modern classroom microscope. In 1672, Leeuwenhoek developed the first simple microscope to view the tiny objects with the magnification of 200 to 300 times. That's why he is called as father of microscopy. The term microscope was coined by the scientist Faber in 1623. This is the image of the microscope, which is compound light microscope. The next one, we are going to see about the parts of microscope. The first and foremost and the important part is the illuminator. This is the light source located below the specimen. The next one is condenser, which focuses the ray of light through the specimen. Then next is stage, which holds the specimen, and objective, the lens that is directly above the stage. The next is nosepiece, the portion of the body that holds the objective over the stage. Then iris diaphragm, which regulates the amount of light into the condenser. Base of the body supports the microscope, which is horseshoe shaped. Then coarse focusing knob, which is used to make relatively wide focusing adjustment to the microscope, in which we can view the objects. Fine focusing knob used to make relatively small adjustment to the microscope. Then next one is ocular piece. Lens on the top of the body tube is called ocular piece, which has a magnification of 10x than normal vision. And this is the difference between the objective lens. There are three types of objective lens in a light microscope: low power, high power and oil immersion. The magnification of objective low power is 10x, high power 40, oil immersion is 100x. Defaultly, the magnification of eyepiece is 10x. So, by multiplying low power with eyepiece 10 into 10, 100x is the total magnification of low power. For high power, it is 450, and then for oil immersion, it is 1000x. Numerical aperture also varies between the objective lens. For low power, 0.25 to 0.30. For high power, 0.55 to 0.65, and for oil immersion 1.25 to 1.4. Mirror used is for low power and high power. concave lens is used. In case of oil immersion, we are using plane mirror. Then focal length approximately 16 mm for low power, 4 mm for high power, and 1.8 to 2 mm for oil immersion. It means that when I will go for higher resolution, focal length will be increased. Next, working distance. Working distance also decreased while going for higher resolution. 4 to 8 mm for low power, 0.5 to 0.7 mm for high power, and 0.1 mm for oil immersion. Maximum resolution for low power is 0.9 micrometer, for high power 0.35 micrometer, and for oil immersion 0.18 micrometer. And this is the image which we can see under light microscope. The first one is the rod shaped bacteria which is called bacilli, and the second one is round shaped bacterium which is called cocci. It has been viewed under light microscope. The next image is the paramecium viewed under light microscope. Thank you.